Hi, my name is Molly Bright, and I'm speaking to you about adding vascular insight into your fMRI experiment and how that can maybe help us better interpret brain function in functional neuroscience. These are my disclosures and acknowledgments. So why do I feel like this is a very important topic for us to be discussing? And fundamentally, vascular function is critical to neural function. There are no oxygen stores in the brain, and when neurons become metabolically active, they rely on neurovascular coupling to supply them with additional oxygen for healthy function. So if you have disrupted or dysfunctional vascular physiology, that will potentially then limit the ability of neurons to function normally. But again, with our community, a lot of us are also relying on things like functional MRI, typically with the intention of using it to map brain activity and therefore neural activity. But fMRI is not a direct measure of neural activity. It is measuring a vascular effect of blood oxygenation level changes. And we need to have an understanding of neurovascular coupling and a model for it in order to infer neural activity from the fMRI time series. So overall, any vascular variability could confound our fMRI estimates of neural activity, or it could actually influence neural activity directly, and that will be the theme of the talk. So where does vascular variability come from? Take your pick. That brain you're interested in studying is inside of a person, and that person has a lot of things going on. And almost all of it could influence your brain signals in some way. But specifically thinking about vascular variability, it is established that lots of these factors, for example, can influence systemic blood flow. And blood flow changes could potentially influence neurovascular coupling on top of that baseline flow level. So usually you may already be familiar with trying to reduce variability in these domains or just document this variability and try to account for it in a group analysis. I'm more interested in vascular variability that happens within one scan session itself. So for thinking about blood, it's kind of natural to first think of the heart. The heart is beating approximately 60 to 100 times a minute, and then that will create this pulsatile movement artifact around vessels and also pulsatile flow patterns. And then the heart rate, that beat-to-beat interval, that's going to reflect a sympathetic, parasympathetic balance, and heart rate changes are associated with changes in vascular tone and therefore the bold signal. But I am more interested in the lungs. Your lungs, again, you're breathing right now sitting in front of me. You're inflating and deflating your lungs maybe 12, 16 times a minute. This will create periodic movement. It will create periodic changes in the B0 field homogeneity, and that will confound your fMRI time series. But what I'm interested in is how changes in breathing and changes in ventilation can actually influence respiration and gas exchange. So changes in breathing will influence the amount of oxygen and CO2 is present in your bloodstream. And that's important because carbon dioxide in particular is a very strong vasodilator. If your CO2 levels go up because of changes in breathing, then you will have widespread vasodilation, blood flow increases, blood oxygen increases downstream, and you will see that in your bold signal. And so that is something we're going to lean on in the rest of this talk. So I'll be emphasizing the effect of breathing on vascular function. Now, many of you may not care. And you see these types of effects as noise, and lucky for you, we have an amazing toolbox of techniques to help you model and remove a lot of these effects. So you often may incorporate a recording through a respiratory belt, a nasal cannula. You have models of how to turn recordings into nuisance regressors in your general linear modeling of the time series, such as retro-ICOR, RVT, or end-tidal CO2 modeling. There are also data-driven, sort of more exploratory approaches that you can draw from, like the tools here. And I'm going to refer you to a course that we ran this weekend, really tackling physiological fMRI signals, how you can model them and remove them or care about them. And really, I'd like you to think maybe this vascular variability, this physiology effect, maybe it's not noise. Maybe we can actually capitalize on these effects and get a new signal of interest that tells us something new about the brain. To do that, well, I usually say you need some sort of variability in that vascular physiology. And like I said, if I just put you in there at rest, there may just be natural fluctuations in your breathing that create natural variability in carbon dioxide levels. In my lab, we often do a little bit more than that. So here's an example of a breath hold task that we use. This would be a type of ventilation challenge. So we give instructions to ask a participant or scan subject to perform short breath hold tasks or maybe paced breathing. And that will cause transient increases in CO2 associated with transient vasodilation and bold signal increases. We could also get a bit fancy. We have the ResBrac system is an example of a gas inhalation system where we can actually target different CO2 levels by delivering breath by breath different gas mixtures through a face mask. And this therefore can modulate CO2 independently of sort of breathing compliance and things like that. So in terms of getting these data, it's certainly easier to just do a resting state scan. It's certainly harder to have all this equipment. But when it comes time to actually characterize the bold fMRI response to these changes, you get what you pay for. When you have a big, robust signal change because of a large, intentional modulation in carbon dioxide, it's easy to model it, pull it out, and characterize it. When you have resting fluctuations, often there are many other phenomena present in your fMRI data around the same frequency, and it's much harder to disentangle. But ultimately, what we try to do is output maps of cerebrovascular reactivity, or CVR. That's the responsiveness of your brain's vasculature to these breathing challenges and CO2 modulations. And we can pull out the amplitude, so how much the vessels dilate, and also the latency, when the vessels dilate. And this produces two interesting biomarkers that are starting to become increasingly used in translational research. So for example, where might we want to bring in this type of information? Well, we know that impaired vascular reactivity precedes and can predict the formation of white matter hyperintensities. And then we think there might be something involved there with vascular contributions to Alzheimer's disease and related dementias. We know that healthy young adults, so in their 20s, no symptoms, but they happen to be carriers of the ApoE4 allele, a risk factor for Alzheimer's, we see widespread reductions in CVR in these individuals. And we also see reduced CVR in multiple sclerosis. It might be related to inflammation, and it can be restored during immunomodulation therapies. On top of that, CVR just declines as you get older. It declines with age. It's lower in hypertension, which many older adults have. So if you're studying an age-related disorder or disease, you may have these effects going on in the background. It may be different between sexes. And it does kind of raise this question. Atypical vascular reactivity suggests potentially atypical neurovascular coupling. So if we don't understand that neurovascular coupling, how can we actually infer neural activity from our bold fMRI time series? And so that is an active question. But I think the first step is to really understand, are there vascular differences in my participants, in my patients, in this broad cohort? And then we'll learn how to better incorporate those effects. My lab has a very practical way to approach this. We know that most people want to do a resting state scan or a task fMRI scan, not just focus on breathing modulations, but we encourage you to try out this hybrid protocol. So here's an example where we asked participants to perform 3 short breath holds prior to a resting state period. You can see that effect in the CO2 recordings, as well as in the bold fMRI time series in gray matter. And we can produce those maps of vascular reactivity, amplitude, and delay from this type of simple protocol. The idea is also that having these modulations lets us model that amplitude and delay better across the entire data set and potentially better denoise some of these CO2-related fluctuations from the resting state period. But I must ask, can we disentangle neural and vascular effects? So here I'm showing an example from a study where we did a working memory test. You can see the block design in blue. And in red, you can see that CO2 levels are entrained to that neural stimulus timing. Breathing patterns correlate with this working memory task. And similarly, you see the same effect here during a hand grip motor task. And you may not know this is happening if you're not measuring. So at the very least, I encourage you to measure breathing changes. But I would also say that breathing is driven by neural activity. And breathing changes neural activity. There are all of these feedback mechanisms that disentangling them may be impossible. So regulation of breathing, you have chemoreceptors in your brainstem and carotid bodies, and ultimately brainstem nuclei can maintain, or trying to change breathing to maintain blood gases within normal limits. There's volitional control of breathing, cortical limbic control of breathing, autonomic control of breathing. I'll point you to these papers for more information. But any task that we ask people to do related to breathing, or any drivers of subconscious breathing changes, probably are involving many areas of the brain in a neural capacity, as well as then influencing vascular physiology. So these systems are very intimately coupled, and disentangling them is difficult. Not only does breathing relate to neural activity, but actually carbon dioxide levels will change neural activity. So in this study led by Ian Driver, we administered gas challenges during MEG scanning. So there's no changing in breathing necessarily or intentionally, but we do have these changes in CO2 levels, which then manifest as changes in alpha, beta, and low gamma power. And we even see that resting fluctuations in carbon dioxide, due to just natural variability in your breathing rate and depth, those also can manifest as changes in neural activity as measured through MEG. Taking this a step further, Tu and Zhang looked at the relationship between breathing rate and depth as measured with this RVT parameter with resting state fMRI time series in rats, but they did it in a very clever fashion. First, they looked at this relationship just in normal resting state with light sedation, and then they began an infusion of sodium pentobarbital to create this isoelectric state, basically silencing neural activity throughout the brain. What they observe is that during this light sedation, where there is neural activity, you see a relationship between breathing patterns and the resting state time series that kind of creates something that looks very structured and network-like in the rodent brain. Whereas in the isoelectric state, that basically disappears. And critically, this RVT measure looked very similar in both of these conditions. So this suggests that there really is a respiration-related brain network that links together aspects of cortex, subcortical regions, and beyond. Now, slightly more controversially, I examined neural and vascular effects simultaneously in healthy adults, administering working memory task, a visual stimulus, and also CO2 gas challenges simultaneously, but with orthogonal temporal designs. And I got these data in several people, multiple scans each, did sort of mass averaging and decomposed them using spatial ICA. And first, I identified components that jumped out that the time series looked very closely related to these neural stimuli, the working memory and the visual stimulus. And the three networks appear spatially as you might expect based on those tasks. But then also in that ICA decomposition, we observe additional networks that are spatially similar to the original three, but their time series looks much more like that gas challenge and driven by vasodilation associated with carbon dioxide changes. This is very similar to work done by Jingyuan Chen at a similar time. Using the Human Connectome project, they modeled the variance associated with this respiration volume measurement, but instead of fitting the data with an empirically derived response function, they actually allowed that response function to be very flexible and used a basis set at the voxel level. And when they combined across subjects in the HCP dataset, looking at just signal in the brain that can be predicted by respiration patterns, they did some clustering, and again, network-like structures appear. And these structures are consistent between two different cohorts, and they cannot be replicated by chance when using breathing from, say, a different subject. And so this adds evidence that there are these structures, long-distance coordinated responses to breathing changes and CO2 changes. So what does this mean? perhaps there are these coordinated vascular networks. And the vascular neural network is a concept that I refer you to this paper here. We shouldn't think about neurovascular coupling at just the microscopic scale. We should really think macroscopically across the brain. So we may see that the vasculature is actually regulated in networks that spatially mirror known neural networks. And this would be a very efficient way to support coordinated neural activity in those systems. But it may also be that breathing and vascular physiology changes can modulate aspects of these neural networks and cause splitting or other phenomena. So finally, yes, neural activity relies on neurovascular coupling. fMRI relies on neurovascular coupling for us to interpret it. And vascular function is critical and perhaps inseparable from neural function when we're looking at the brain. So going forward, I encourage you to try more multiparametric fMRI to achieve insight into neural and vascular properties. So basically, try to enjoy and embrace the ambiguity and duality of fMRI signals. There are also many concrete approaches to characterize breathing and really go after the associated effects in your data. And then finally, we may need to just continue to embrace multimodal neuroimaging to better discern neural and vascular contributors to the phenomena we observe in our neuroimaging data and any potential pathology. I'll leave you with a few pointers towards resources that may help you along these lines. And with that, I'll thank you for your attention. And anything interesting that I'm doing is the work of these very amazing people that I get to work with on a daily basis. Thank you very much.